So good day everyone. Welcome back to Magic School. Today we'll be going into the translation of mRNA, biosynthesis of proteins, and then this is under our molecular biology course. So yeah, let's get into it. So now the biosynthesis of proteins. In the simplest sense, expressing a gene simply means manufacturing its corresponding protein. And this multi-layered process has two or more steps, which are the transcription and translation. Now the first step is actually this transcription. The first step is transcription, which is the formation of RNA from DNA. So how this happens is the enzyme, the helicase enzyme, and then unwinds the DNA until it reaches a particular site, and then it stops, and then the RNA polymerase comes on to synthesize the messenger RNA or the pre-messenger RNA. Now this pre-messenger RNA is spliced to form the mature mRNA, and this splicing involves the removal of introns, and now the mRNA is ready to be translated into proteins. So now after the transcription, the translation begins and the mRNA moves out of the nucleus into the cytoplasm and gets to the ribosomes. So now for this synthesis, for this translation, there are actually 5 stages. The first stage is the activation of these amino acids. Now the amino acids are present in the cytosol and they have to be activated. So the carboxyl group of amino acid is actually activated, and now a link has to be established between the amino acids and the mRNA, and this link now is gotten by the tRNA. So now the tRNA covalently binds to is covalently linked to this amino acid, and this occurs in the cytosol, and ATP is actually used in this process. And also there's an enzyme, amino acyl tRNA synthetase, and it is actually magnesium dependent. So here the right amino acid must actually be attached to the right tRNA. So now the second step, which is initiation. So now the mRNA binds to the ribosomal subunit, both the larger and the smaller subunit, to form the initiation complex. Now the initiating amino acyl tRNA base pairs with the codon AUG that signals the beginning. This stage requires GTP and initiation factors. So here the tRNA binds to the amino acid methionine, because AUG is the codon that signifies for methionine. So now the tRNA binds to methionine, and then this tRNA binds to the first codon, which is the start codon, and start codon is AUG on the amino acid, and binds there on the amino acid, and this amino acid is called the initiating amino acyl tRNA. Now the third stage is elongation. So now after the first tRNA has been bound to the mRNA, now the next tRNA that is corresponding with the next codon on the mRNA then comes in and binds at the A site, of the ribosome, and then peptide bond is formed between the two amino acids. So now, and this continues, and more amino acids are linked together, So let's look at the diagram. The first step in elongation is decoding. So now we see that there's an incoming amino acyl tRNA that binds to the A site. So now a peptide bond is then formed between the 3 and 4 amino acid. That's where we see the second step, peptide bond formation. Now 3 and 4, there's a peptide bond now between them. Now the ribosome moves, that is translocation. Now as the ribosome moves along the RNA, the tRNA labeled 3 there moves to the E site, and then the one that was in the A site moves into the middle. Now eventually the tRNA that is labeled 3 diffuses away back into the cytosol and binds to amino acid, and then it continues on and on. Then another tRNA is brought into the A site, and a peptide is now formed again, amino acid labeled 4 there, and it goes on again. Now something to note here is that the tRNA that is coming in must correspond or is complementary to the codon that is base pairing with on the mRNA. So like we see, for example, the AGU on the RNA, that is the first one, binding to the UCA on the tRNA, so it's complementary. Just how we have in the DNA that G is complement to C and A corresponds to T. So also here, that is how it works, U corresponding to A and then G to C, and that's how it also works here for the translation. Something to also note here is that the ribosome reads the mRNA from the 5' to the 3' direction. Now, the coming in of the amino acid tRNA and the movement of the ribosome is facilitated by the hydrolysis of GTP. GTP is being used as a source of energy, and it's also facilitated by the elongation factor G. The fourth step in translation is termination. Now, the translation goes on until a stop codon is reached. And we have 3 stop codons. We have UAG, UAA, and UGA. Now, a simple way to be able to remember all of them is to know that all the stop codons start with U. Now, the first stop codon is UAA. Now, the next one is UAG. Now, the last one, you just bring the G first and then put the A at the back, so it's now UGA. So just to remember, all of them start with U. No one starts with G or no one starts with A, and that way it's quite easier to remember. Now, upon the encounter of a stop codon, the tRNA does not recognize this stop codon, so in place of this tRNA, the release factor or a release factor binds and facilitates the release of this mRNA and the release of this newly formed polypeptide chain now, and then the ribosome is recycled again for another round of translation. Now, the fifth stage is the folding and the post-translational processing of post-translational modification. So now the protein or the polypeptide chain that was just formed is then folded in 3 dimensions to achieve its tertiary structure. So now it also undergoes modifications before or after the folding. It could be addition of phosphoryl groups, addition of acetyl groups, methyl group, or carboxyl group, and then depending on if it was synthesized as a zymogen, it can then undergo proteolytic cleavage. These modifications also can include the removal of one or more amino acids, and also include the attachment of oligosaccharide or prosthetic groups in the case of proteins that actually that function as enzymes. Now, these modifications can affect the protein stability, their localization, and their activity in the body system. Now, in the process of this translation, there are 3 types of RNA that are being used or are seen. We have the messenger RNA, the ribosomal RNA, the transfer RNA. Those are the 3. So now the ribosomal RNA is part of the ribosome. Now the messenger RNA is a single-stranded copy of the gene or single-stranded copy of the DNA, and then the transfer RNA is what binds to amino acids and bring them to the ribosomes to form the proteins. Something to note about translation is the formation or the synthesis of proteins, not the synthesis of amino acids. So the mRNA just brings about the arrangement or brings the code of how these amino acids should be arranged to synthesize a particular protein. And lastly, for today's video, as I said before, the start codon is AUG and the stop codon there are 3 of them: the UAA, the UAG, and the UGA, and then the signal the end of translation. So that is the end for the synthesis of proteins for today, and next we'll be going into inhibition of protein synthesis. Thank you, and don't forget to like and subscribe. Thank you.